Hello everyone, and welcome to today's webinar, Understanding How In Vitro Macrophage Assays Drive IO Drug Development Forward. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotech. To learn more, visit miltenyibiotech.com. Let's get started. Before we begin, I would like to remind everyone that today's event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. Thibaut Janss, Senior Scientist at ImmunXperts and Nexus Group Company. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Janss, you may now begin your presentation.
Hello everyone, and welcome to this webinar. I'm Thibaut Janss. I'm a Senior Scientist at ImmunXperts, a Nexus Group company. So first, I would like to give you a bit of a background about the company history. So ImmunXperts was founded in 2014 by Sophie Patain together with Felicitas and Arnaud Grominger. And first, the company was focusing on in vitro immunogenicity assay. Then in 2016, with the booming of the immunology development and, for example, the checkpoint inhibitor field, the company expanded to in vitro functional and potency assay for higher drugs. In 2017, we moved to new dedicated facilities, and we currently have more than 17 colleagues. We have been working on 200 projects for more than 70 customers in 18 countries. In March this year, we have been acquired by the Nexus Group, a world-leading provider of services with more than 20-year experience in the infectious disease field. Our expertise is in vitro immunology in a R&D context, but at the highest level of quality. We work with all types of products: antibodies, cell therapy products, RNA and DNA vaccine, viruses including oncolytic virus, and small molecule. We have a recognized expertise, and we are involved in 4 international R&D projects funded by the European Union. It takes quite a long time to get a market authorization for the drug. It's starting with the target selection, working on genomics and proteomics, then lead discovery with assay development, high throughput screening, medicinal chemistry with library development, medical chemistry, and in silico screening, before moving to in vitro screening and in vivo screening, before the first in many applications in clinical trials that will hopefully lead to the commercial acceptance of the product. We believe that with high quality in vitro screening, you will be able to move faster from in vitro screening to clinical trials and also to reduce the use of animals. You are probably familiar with this nice scheme from the chain paper, showing the complex interaction that can occur in the tumor microenvironment between the immune cells and the tumor cells, but also with the different immune cells together. At ImmunXperts, we have different assays focusing on T cells and myeloid assay. Today, our interest will be on myeloid assay and more specifically on macrophages assay. Macrophages are phagocytes that serve as first line of defense against pathogenic intruders. These innate immune cells mount pro-inflammatory response to pathogen and repair damaged tissue. The diversity of the macrophages in vivo is quite important. We can find different types of macrophages in the different organ and tissue with different function. So it's really an important family with a lot of different types of macrophages with different function. Classically, in the literature, we can find a classification, a simplification of this classification with M1 and M2-like macrophages. The M1 macrophages inhibiting the tumor growth or the pathogen invasion, and the M2 macrophages being involved in the healing process. The M1 macrophages secrete INOS, pro-inflammatory cytokine, express high level of MHC2 and co-stimulation molecule. They are also more linked to the Th1's response, while the M2 macrophages secrete high level of arginase, cytokine like TGF-beta and VEGF, and express CD206, and other markers like CD100 and 63. And they are also considered to be involved more in the Th2 response. The simplification of the classification of the macrophages is quite useful in vitro, because it corresponds to the type of macrophages that you can induce in your assay. So the M1 macrophages, which depend on the stimulation of interferon gamma and LPS, and which are involved in Th1's response, as I mentioned earlier. In the M2 family, you can find three subclasses of macrophages. First, the M2A then can be induced with IL-4 and IL-13, which are involved in Th2 responses like allergy. Then the M2B that can be induced with immune complex and TLR ligand, which are also involved in Th2 activation and in immunoregulation. And finally, the M2C, which are induced by IL-10 and which are involved in immunoregulation and in tissue remodeling. These M2C are often used as a surrogate for the tumor-associated macrophages. You can see on the right on the slide that these different macrophages are activated through different pathways, through the different transcription factors. For an example, the interferon gamma can induce the STAT1 and interferon responding factor pathway in the macrophages, while on the M2, for example, you have the IL-4 that will induce the phosphorylation of STAT6 and the activation of the gene depending on this transcription factor. An important notion to know about the macrophages is the plasticity of the macrophages. This concept has been historically found in the literature, mentioning that macrophages can be switched from a phenotype to another one. Then an M1 macrophages can become an M2-like macrophages, and then an M2-like macrophages can become an M1-like macrophages. Recently, a more complex model has been proposed to investigate different type of macrophages, which is related to the ontogeny, and mentioning that it's not so much plasticity of the macrophages that we can observe, but different recruitment of different origin that will lead to a more complex description of the macrophages. Currently, literature we find paper defending both models. One that stands that macrophages can be found in different states, the cells can be found in different states, and another one saying that it's different cells are different origin, resulting in different recruitment at the tumor sites, for example. So to look in the tumor microenvironment, so you can find macrophages, so the TAMs, the tumor-associated macrophages. Usually, these macrophages has immunosuppressive property and also favorize the tumor growth through angiogenesis. The goal of some therapeutics is to repolarize or to change these macrophages to a more pro-inflammatory phenotypes that will induce immunostimulation, an increased antigen presentation, and a T-cell activation, leading to a tumor regression. There is a lot of different mechanisms that can be targeted to re-educate the macrophages and go from this immunosuppressive phenotype to a more pro-inflammatory phenotype. So, for example, you can use CD40 agonist to trigger the expression of pro-inflammatory cytokine, or just use PD-1 or PD-L1 blockade to interfere with the immune suppression mechanism used by the macrophages to suppress the T-cells response. There is a lot of different mechanisms that can be targeted by the macrophages. You can see on this slide that there is a lot of molecules that are currently under investigation, currently tested in clinical trials. Today, we will focus on the drug that can affect the polarization of the macrophages or their interaction with the T cells. So we will focus on three types of assay. The first one is the M1, M2-like macrophages polarization assay, which can be used to assess the effect of test compound on macrophages polarization, or to assess the effect of a test compound on macrophages phenotype and function. The first one is a model that study the effect on the macrophage recruitment and differentiation, and the other one, the effect of a test compound on the already differentiated and activated macrophages, and so that will interfere with their function. To use model that another model is the M2-like suppressive assay, which allow us to assess the effect of a test compound on the suppressive properties of M2-like macrophages and their interaction with the T cells. And the latest assay that we will be discussing today is the phagocytosis assay, where we can assess the effect of a test compound on the macrophages' phagocytic activities. But to perform such assay, it all starts with high quality cells. So happy cells mean happy assay, like it is shown in this nice scheme from Miltini. So the quality of the primary cells means that you will reduce the variability and increase the reproducibility of the results. The quality means that you need a high viability and high functionality of these cells that are the most critical reagent in your assay. To ensure this high quality, you need to use standardized procedure for sampling, shipping, isolation, cryopreservation, tolling, and handling of the cells. To assess the functionality of the primary cells and so the quality of the primary cells, at ImmunXperts, we use a stimulation over 7 days of the PBMC with a polyclonal stimulation, so an agonist of CD3, or with a naive antigen, so the keyhole lymphet hemocyanin in this case. After the 7 days of culture, we are measuring the proliferation of the CD4 T cells and the CD8 T cells to assess the responsiveness of the cells. This proliferation is measured with the incorporation of EDU, and the readout is performed by flow cytometry with the MAC content. You can observe that when non-stimulated, there is a very low percentage of proliferating cells. Then when stimulated with KLH, we observe that more than 2% of the CD4 T cells are proliferating, meaning that the cell preparation has functional cells, and so that these cells can be used in downstream assay. Another important part of our QC procedure to assess the quality of the PBMC is also a subpopulation analysis to determine the different proportion of the different subpopulation. It can be quite interesting because, for example, the percentage of monocytes can vary from one donor to another. And for myeloid assay and macrophage assay, we will select donor with a higher percentage of monocytes, meaning that we can have more cells and perform more condition or more assay with the same donor preparation. So to work with macrophages, it all starts with monocyte isolation. The monocyte can be isolated from different sources, from cryopreserved PBMC, but also from whole blood or leukapheresis. The leukapheresis can be quite interesting for myeloid assay because they usually present higher percentage of monocytes than in a PBMC from whole blood. To isolate the monocytes, you can use different kits using positive or negative selection. In the result that I will present in the following slide, we only use CD14 positive selection with the Miltini bits to isolate the monocytes and induce their differentiation into macrophages. To ensure the quality of the assay, I will say it's quite important to follow the kit recommendation. So an incubation from 2 to 8 degrees means an incubation in the fridge, not on ice. It will allow you to increase the purity but also the recovery of the cells. At ImmunXperts, we also use pre-cooled tube for the harvesting of the cells to avoid that the macrophages stick to the tube and so to lose a part of the macrophages or the monocytes. And also it's important to use cold medium after the isolation. So once the monocyte has been isolated, you can start proceeding to the macrophage differentiation. For the M1-like macrophages, we use 6 days of GM-CSF followed by 2 extra days of stimulation with interferon gamma. For the M2-like macrophages, it's 6 days of MCSF followed either by 2 days of IL-4 together with IL-10 and TGF-beta to induce M2C, or IL-4 and IL-13 to induce M2A. Once this macrophages has been differentiated, you can assess the version I by flow cytometry by using different marker characteristic of the M1 or the M2-like macrophages. It is important to know that the macrophages are quite delicate cells, so the culture condition has to be properly set up. This is what the M1 or the M2-like macrophages can look under the microscope after the full differentiation. The M1-like cells are highly adherent. They present long cytoplasmic extension. There is also a high risk of the medium exhaustion, meaning that you will have to refresh several times the medium during the differentiation process. And the yield is about 15 to 30%, so meaning that you recover 50 to 50% of M1 from the monocytes that you have been seeding. The viability can be also quite tricky with the M1, but I will come back to this on the next slide. With the M2 macrophages, they present a low adherence, small cytoplasmic extension. The medium exhaustion risk is lower than with the M1, and the yield is also better with these cells. So the M1 are highly sticky cells, and it can be quite tricky to get viable and functional M1 after the differentiation with them from monocytes. In this slide, it's an experiment where we try different protocols to harvest the macrophages. So either an enzymatic method with acutase, non-enzymatic buffer, or just scrapping of the cells. We also seed 1 to 4 million cells in a 12-well plate. You can observe that if we use the scrapping method, we have a low viability of the macrophages. If we use on the other side the acutase, we have a higher percentage of live cells, and it is similar between the different cell density tests in this assay. With the non-enzymatic buffer, the viability increases with the cell density and is higher than with the scrapping method. For the yield, there is less variation between the different cell density, but you can observe that with the higher cell density, the non-enzymatic and the scrapping method present better result than with the lower cell density. But this cell density can also affect other parameters. If we look at CD86 expression on macrophages, you can see that the lower the cell density is, the higher the CD86 expression is. So meaning that this cell density has also an influence on the quality of the macrophage that you can recover and the expression of some membrane marker, but also the production of cytokine. It's also important to note that the enzymatic method can affect the phenotype of the macrophages. And if we look at the CD209 expression, the acutase induce a decreased expression of this marker. It's why we usually try to avoid to use this enzymatic method to harvest the macrophages. So it's really, you have to, depending on the culture condition, to take the better compromise, the best condition to get live cells, functional cells, and expressing the right marker at the right level. So it's quite complicated with the macrophages. And at ImmunXperts, we usually use the non-enzymatic buffer to harvest the macrophages and the cell density from 2 to 3 million cells per well in a 12-well plate. Once you get these macrophages, you still have to work with them, but also to characterize them. The flow cytometry is quite an important tool to characterize the macrophages, but these cells are highly autofluorescent, meaning that you have a high signal with non-labeled cells in the UV, violet or blue laser, as you can see on this dot plot. Then it's quite important to use the relevant control, so the fluorescence -1, to be sure of the background staining or the background autofluorescence of the macrophages and to select the right positive signal. The reantibodies from Miltini can really help in this case because they will increase the resolution. The monocytes in the macrophages also express CD16, CD32, and CD64, meaning that they are more susceptible to the specific staining with the test antibody. So you have to use FC block or the reantibodies that will allow you to get rid of this problem. Note that we know how to get these macrophages. We will go further in the assay that we can perform with them. So in the M1, M2 polarization assay, we can perform the the assay in two configurations. In the first one, the test compounds are isolated from the monocyte isolation step to assess their effect on the differentiation process. But when the macrophages are fully differentiated, we can harvest them, wash them, and reseed them in presence of the test compound to assess the effect of the test compound on the function and the phenotype of the macrophages. So this is an example of the kind of result that we can obtain with an analysis of the cell surface marker expression on M1, M2A, and M2C. If you look at the expression of CD163, a marker that is characteristic of the M2-like macrophages, you can observe that we only have an expression in the M2-like macrophages, and this expression is higher in the M2C. CD209 is another marker used to characterize the M2 macrophages, as it is expressed by M2A and M2C, but very low expression is detected on M1. CD206 and CD210R are expressed by both types of macrophages, but usually the expression is higher in M2-like macrophages. For the M1-like macrophages, we usually look at the co-stimulation molecule, but you can also look at the MHC class 2 expression to check these macrophages, because the expression of this marker is usually higher on M1 than on M2. You can also observe that the M2A are between the M1 and the M2C for the expression of co-stimulation molecule. And finally, we also like to check the PD-L1 expression, as we can use it as a control in some on our assay, and it is expressed by all types of macrophages, but usually the expression is higher in M2C, which are the most suppressive macrophages. In this example of a vaporization assay, we treat fully polarized M1, M2A, or M2C with interferon gamma and LPS. On the cell surface marker, we don't see so much variation. We mostly observe an increased CD86 expression on the M2A and M2C treated with interferon gamma. And if we look at the cytokine production below, you can observe that IL-10 is not produced by M1. We find a production in the M2A and M2C macrophages. And this expression is decreased in the cells treated with interferon gamma. If we look at the IL-12 P70, as expected, it is produced by the M1-like macrophages, a little bit by the M2A, but no production is detected in the M2C condition. But with the IL-12 P70, we don't see so much variation with the different treatment that we use in this assay. So this is the example of the repolarization assay, and I will come back later with an example of a polarization assay where we use a test compound that has been published in the literature. With these macrophages, we can also perform a suppressive assay, so to assess the suppressive effect on the macrophages on the CD4 T cells. This assay can also be used with CD8 T cells or PANT T cells, but today I will only discuss examples involving the CD4 T cells. So what we are doing is still the same protocol to induce the differentiation of M1 or M2-like macrophages, and then performing culture of these cells with autologous CD4 positive T cells in presence of the test compound and a CD3/CD28 agonist to induce an activation of the T cells. After 5 days of co-culture, we can measure the proliferation of the cytokine production from the CD4 T cells. It's important to note that the macrophages has a phagocytic activity. So if you use beads coated with agonists of CD3 and CD28, you can observe a suppression of the response just because the macrophages will phagocyte the beads. So it's better to use a soluble activator of CD3 and CD28 in this kind of assay to induce an activation of the T cells and be able to assess the suppressive activity of the macrophages. We also usually use a ratio of 1:5, so one macrophages for 5 T cells. To assess the T cell proliferation, we use a proliferation dye, so we prefer to use a violet proliferation dye because they have a lower toxicity and also allow to have a higher resolution, as we use the replication index as a measure of the replication of the T cells. It's also important to include a viability dye in the readout to get rid of the dead cells. And for the CD4 staining, it's better to use fluorochrome that will allow you to discriminate the CD4 T cells from the macrophages, so to avoid the problem of the autofluorescence of the macrophages that will contaminate the CD4 response. So on most cytometers, you have a red laser, and so the APC can be a good choice for the CD4 labeling and to discriminate the CD4 T cells from the macrophages. So this is the kind of result that you can get with a macrophage suppressive assay. If we look on the right at the cytokine secretion, so for the interferon gamma, you can observe that we have a high secretion of interferon gamma in the CD4 T cell alone, and a similar level in presence of the M1-like macrophages. In presence of the M2-like macrophages, we have an important decrease of this secretion, meaning that the macrophages suppress the T cell activation. If we look on the left at the proliferation, you have in gray the level of the CD4 T cell alone. You can observe that with in presence of the M1, the proliferation is slightly higher, but we have an important decrease of the proliferation in light blue in presence of the M2 macrophages. And you can see that in this example, the proliferation stops after 2 cycles of replication, while we go up to 6 or 7 replication cycles in the CD4 T cell alone or in presence of the M1-like macrophages. So meaning that we were able to suppress the activation of the CD4 T cells. With these 2 assays, we perform a case study with LILRB2. So this is a publication from 2018. And so in this publication, using mostly mouse model, they show that if you block LILRB2, you can prevent the differentiation of monocytes into M2-like macrophages, but that this LILRB2 can also act as a checkpoint inhibitor specific of the myeloid cells. So we do 2 assays using this blocking antibody for LILRB2, first a polarization assay and then a suppressive assay. So in the polarization assay, we had the anti-LILRB2 antibody from the monocyte isolation step. And this is the kind of result that we obtain. So if we look at CD163 and CD209 expression, 2 markers which are characteristic of the M2-like macrophages, in the control condition, you can observe that in the first donor, we have a purity of the M2 of 86.7, and in the second donor of 92.5. In presence of the LILRB2 antibody, we decrease this percentage to 49.8 in the first donor and to 68.6 in the second donor. So meaning that the addition of this test compound allows us to prevent the differentiation of monocytes into M2-like macrophages, and that this assay can be used to assess the effect of a test compound on the macrophages polarization. Then, with the same test antibody, we perform an M2 suppressive assay, but in this case, the LILRB2 antibody was only added during the co-culture of the macrophages with the T cells, so not from the monocyte step, only during the co-culture. And so, as usual, we perform a QC of the macrophages to check that we have nice M1 and M2-like macrophages, that they express the right marker, and it was the case of, for example, an expression of CD163 only M2-like macrophages, while the co-stimulation molecule were highly expressed on M1-like macrophages. So we measured the interferon gamma secretion after 5 days of co-culture. You can observe that we have high secretion in presence of the CD4 with the CD4 T cell alone stimulated with the CD3, CD28 agonist. In presence of the M1 in the 2 first donor, we don't have a significant effect, and in the third donor, we have a slight decrease, but that is that was caused by an exhaustion of the medium. So the medium was completely exhausted, and so it decreased at the end the secretion of interferon gamma by the CD4 T cells. If we look at the M2 condition, so in this case we don't have an exhaustion of the medium, but we have an important decrease of the interferon gamma secretion in comparison to the CD4 or CD4 plus M1 condition. In the co-culture, we also test 3 antibodies so the Opdivo, so clinical anti-PD-1, the LILRB2 antibody, and an anti-PD-L1. And with the 3 antibody, we were able to increase the interferon gamma secretion in the CD4 plus M2 co-culture, and so to interfere with the suppressive property of the macrophages. If we look at the proliferation in the same assay, we have a similar effect of the CD4 and CD4 plus M1, so 100% proliferation, sometimes slight decrease because of the exhaustion of the medium. But with the test compound, we don't see so much effect. Just in donor 2, a significant increase in presence of the LILRB2 antibody, and in the donor 1, a trend to increase, but not significant in presence of the LILRB2 antibody. So altogether, these results show that this assay can be used to assess the effect of a test compound on the suppressive function of the macrophages, so that this suppressive assay can be used to screen molecules that are targeting the suppressive mechanism used by the M2-like macrophages to suppress the T-cell activation in the tumor microenvironment. The last assay I will discuss today is the ADCP assay, so antibody-dependent self-phagocytosis assay. So in this assay, we induce macrophage differentiation, and then we perform a co-culture of these macrophages with the target cells in presence of the test compound. And we can measure the phagocytosis of the target cells by the macrophages. In the following slide, I will present the result based on flow cytometry readout. But we installed this week the first InQSite A65 in Belgium, and we will also adapt this assay with InQSite readout. And so in the test study that we perform at ImmunXperts, we perform M2C differentiation, so MCSF, then IL-4 and IL-10 and TGF-beta treatment. We co-culture with these macrophages with the target cell, which in this case are B cells, so CD20 expressing cells labeled with a proliferation dye. And then we use the rituximab as a positive control, so an IgG1 anti-CD20 antibody, so targeting the B cells. And for the readout, we use the flow cytometry, so as the target cells are labeled with a proliferation dye, and if we use the CD11B as a marker for the M2 macrophages, it means that the double positive cells will be the macrophages that has performed the phagocytosis of the target cell lines. So this is the kind of result that we can obtain. So you have the macrophage only with no phagocytosis as expected. With the human IgG1 isotype control, we trigger some phagocytosis activity by the macrophages, which is expected as this antibody will increase the activity of the macrophages through the Fc receptor present on the macrophages. And then with the rituximab, so the anti-CD20, we increase the percentage of phagocytosis because we also increase the specificity of this phagocytosis thanks to the antibody targeting the cell lines. Meaning that this assay can be used to assess the effect of molecule interfering with the phagocytosis property of the macrophages, and for an example with IgG1 form of antibodies. So with all the assays that I present today, you can screen a lot of different mechanisms, study a lot of different mechanisms and molecules targeting these mechanisms that can have an important function in the tumor microenvironment. Moreover, all these assays have been developed with human cells and so are more representative to the human system as overall system can be, for example, the in vivo assay using mouse. I would like to finish by a few words about the Nexus group. So ImmunXperts, as I mentioned earlier, joined Nexus group in March this year. The Nexus group has different facilities in Europe, in Canada and the US, with different laboratories, with different capacity. So for example, in Canada, in Laval, they have BSL2, BSL2 plus laboratories and animal facilities with a focus on infectious disease and immuno-oncology. The Seattle site has state-of-the-art laboratories with a focus on metabolic disease and oncologic disease. We also have a partnership with Public Health England with access to BCL2, BCL3, and BCL4 laboratories and animal facility in the leadership of world-class scientists. And so through all these different sites and all these different capacity, the next list has services in lead optimization, in vivo screening, in vitro screening, protein science, and clinical trials and large-scale advanced assay, meaning that we can help you from the discovery and early discovery development to the phase 3 clinical trials. If you wish to receive more info, don't hesitate to contact us on the email address info@immunexperts.com. I would like to thank you once again for attending to this webinar, and I will be happy to take any question. Thank you very much.
Thank you, Thibaut, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. Okay, let's get started. Our first question is, do you culture your cells under physioxic conditions? If not, why not?
No, that's not something we are doing, and that's not something we have done with the data present today. That's something we have tried with a specific incubator called the Avatar that allows to cultivate the macrophages in the hypoxic condition. It was giving nice results, but the issue with this kind of system is that most of the time you have really a limited number of plates that you can cultivate in this system. And so for screening purpose and to screening a large number of molecules and test conditions, that was not fitting our application. So it's why we choose to, for the moment, to stick to the regular incubator.
Okay, thank you. And let me ask you another question here. Do you remove beads before use of cells and assays?
No, with the Miltini beads, it's not needed because they are biodegradable and not toxic, so they will quickly disappear from the culture medium. But if something that's before you, there is also some negative selection kit available for the isolation of the monocyte, and we have tried them in our assays, also working quite well to generate the macrophages.
Okay, next question. Which microscopy is used and what magnification for M1 and M2 morphology?
Yeah, so on the picture I show today, it's a classical contrast phases microscope that we use, and I think it's a 200 time magnification, but I would have to confirm that, but I think it's a 200 time magnification that we use in a classical microscopy.
Okay, thank you, Thibaut. Next question. Which non-enzymatic buffer do you recommend?
So for the harvesting of macrophages, we usually use a PS-EDTA-based buffer, and for a specific reference, we use the Versin buffer for the harvesting of the macrophages that I present in this webinar.
Okay, and our next question is, what ratio of macrophage to CD8 T cells do you use?
So the one that we have tested is the same for PAN T cells, CD4 T cells, and CD8 T cells. So it's still a ratio 1:5. So 1 macrophages for 5 T cells, and it gives similar results in the different with the different subpopulation of T cells.
Okay. Next question. Do you prefer coated anti-CD3 or soluble anti-CD3 to stimulate T cells in a suppressive assay, and why?
Yes, we prefer soluble anti-CD3, and for two reasons. So the first one is that from assay to assay, you have less variation with soluble CD3 because with the coating, it's a quite tricky step that can trigger some variation from assay to assay. So you're not always sure to have scored the same amount of antibody. And the second thing is that we usually seed first the macrophages on the plate before the T cells. And so there is a risk that the macrophages, by sticking to the plate, will hide the CD3 antibodies for the access to the T cells. So it's why we usually first seed the macrophages, then the T cells, and then we had the CD3, CD28 agonist to activate the T cells.
Okay, next question. Do you have a positive control for macrophage polarization or suppression assay?
So yes, so it's a control we show in the different data in the webinar. So for the polarization and the suppression assay, we can use the anti-LRB2 antibody, so that will act both as a checkpoint inhibitor, but also as an inhibition of the M2-like macrophages differentiation. And in the suppressive assay, you can also use classical checkpoint inhibitor like anti-PD-1 or anti-PD-L1 that will also have a nice effect on the interferon gamma secretion by the T cells.
Okay, thank you so much. The next question. Do you look for secreted interferon gamma or intracellular interferon gamma by flow?
So we look to secrete interferon gamma. So in this data set that we present today is by ELISA, and it's for the same reason. So it's easier for screening purpose to look at the secreted interferon gamma than to the intracellular one. And we compared the macrophages alone and the macrophages in co-culture with CD4 T cells to be sure that the main source of interferon gamma in our assay is the T cells and not the macrophages. And usually we have some secretion of interferon gamma with the M1-like macrophages alone, but it's very low in comparison to the co-culture condition.
All right, we've got some really great questions here, a lot of good questions. Do you spike in test compounds again during the course of the assay?
So in the polarization assay, when we are refreshing the medium, we add again the test compounds. So for example, with the LRB2, when we are refreshing the medium or adding new cytokine, we also add the antibody again. But in the case of the suppressive assay, we don't spike the test compound during the assay. So it's just added at the beginning of the co-culture, and then we don't add it anymore.
Okay, thank you. Next question. Have you tried inducing tumor-associated macrophages using tumor-conditioned media?
No, that's not something we have done yet, because in our application we need to have quite stable and reproducible results. And this kind of tumor-conditioned media can induce more variation in the assay. So for the moment, we have only stick to the cytokine cocktail differentiation.
And the next question. Have you tried the combination treatment of Opdivo and anti-LILRB2 on macrophages?
No, that's not something that we have done yet, but we think to include it in our next essay to see if there is a synergic effect between these two compounds at different targets targeting different mechanisms. It could have a nice synergic effect, but we don't know yet.
Okay. Do you have experience with other immune cells like microglia?
No, it's not something that I have experience with.
Okay. What are the polarization efficiencies?
So for the M1 and the M2-like macrophages, when we are analyzing the cell after the 8-day differentiation process, in the control condition, depending on the donor, we are between 80 to 95% of M1 or M2 cells. So it's a quite efficient differentiation process.
And I have a few more questions here. The donors you talked about, were they humans or mice?
No, it's all the data I present today we were with human donors, so with PBMC from human and then the monocytes from a healthy human donor.
Okay. Did you try to upsell to harvest macrophages? Excuse me.
Yes, that's something that we have tried. It is increasing the yield and the number of macrophages that you can get. But what we observed is for the M1-like macrophages, the expression of the co-stimulation marker was less important on treated with the upsells, and they were not working so nicely in the suppression assay as control. So we prefer to go back to the regular plate, even if the yield and the viability is a bit lower with this regular plate.
Did you try to stimulate T cell with MODC in the suppressive assay?
That's something that we haven't done yet, but that we have considered to do a few times, but we haven't had the opportunity to do it yet. But I believe it should work to use CD4 T cells or CD8 T cells from one donor, the macrophages from the same donors, and the DC from an allogenic donors to induce an activation of the T cells. It's just then the checkpoint inhibitor like PD-L1 can be also expressed by dendritic cells, so it might also have an influence on the assay, and the increase observed might be also due to the DC action and not only to the macrophages action. It's why for the moment we only stick to models with macrophages and T cells to be sure that we are targeting the molecule expressed on the macrophages and not on another cell type.
Okay, thank you. Next question. Is there an upregulation of some M2 markers while polarizing macrophages to M1 and vice versa?
In a repolarization, it's possible. So for an example, with some test compound like the LPS that will increase the expression of some M1-like marker like the CD86 or the CD80. In some donor, you can also increase the expression of CD163 or CD206. So it's possible, and it's why it's also important to look at the function of the macrophages and, for example, to the cytokine profile secretion. So that will help you to understand if it's trigger a more, I would say, inflammatory or anti-inflammatory polarization of the macrophages.
Okay, we've got one more question here. Mature macrophages do show vacuoles. I could not see that in your images that you showed. How long were they cultured with cytokines?
So with the activation marker, it was for 48 hours. So if we look at with a bigger magnificent, we can probably see this structure, but what we mostly observe at first view with the maturation of the macrophages is the increase of the dendrite and the cytoplasmic prolongement, and also of the cytokine secretion and the exhaustion of the medium because they become quite active.
Okay, and we have another question that just came in. Have you generated macrophages from frozen PBMCs, and if yes, how did they behave compared to fresh? And did you add differentiation cocktail right away or one day later?
So in all the data I present today, it's macrophages generated from frozen PBMC. We have compared in a few assays the frozen versus the fresh PBMC, and we didn't see so much difference in the assay shown today. And we always directly had the cytokine. So we directly isolate the macrophages and then put them in culture with the cytokine. We never try to wait one day before adding the cytokine to induce the differentiation of the macrophages.
Okay, thank you, Thibaut. Looks like we don't have any more questions. Do you have any final comments for our audience?
Not. I will just thank everyone for attending to this webinar and for the interesting Q&A session. So thank you very much.
And thank you again, Thibaut, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Miltenyi Biotech, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.